Hello, my name is Joey Peters, and my project is regulation of the BMP receptor WIT in Drosophila melanogaster. So our lab studies neuronal development in fruit flies, specifically at the neuromuscular junction. This is the region where motor neurons innervate skeletal muscle cells. The amount of growth, or lack thereof, is quantified by the number of terminal axons, or boutons. My project involves studying a specific gene called WIT that we believe acts in a signaling pathway responsible for neurodevelopment. My hypothesis is that tricornered, a gene previously identified by our lab, phosphorylates WIT to inhibit synaptic overgrowth. To study this hypothesis, we needed to make two mutated WIT constructs, one that acts as always phosphorylated and one that acts as never phosphorylated. To generate these, we had to put an @P scar into the WIT gene. To do this, we utilized CRISPR-Cas9 technology. CRISPR-Cas9 is essentially a cut and paste mechanism. We make a guide RNA that complexes with the Cas9 enzyme, telling it exactly where to cut in the fly DNA. Then, in homology-directed repair, a piece of DNA that we make, which is called a homology-directed repair template, is pasted into the cut. This piece of DNA is what contains the @P scar. So in order to do CRISPR-Cas9, we had to make our guide RNA, and we had to make our homology-directed repair template. To generate the HDR template, 4 distinct DNA segments were amplified by PCR: a left WIT flank, a right WIT flank, a plasmid backbone, and an insert containing DS-RED, ampicillin resistance, and the @P scar. We'll get to DS-RED later. These were then confirmed by gel electrophoresis, and then using specific primers that we designed, their overlapping ends were annealed and ligated in an assembly reaction, generating a working plasmid. This was then transformed into bacterial cells to be replicated. To make the guide RNA, a site-directed mutagenesis reaction was used. This generated a tiny substitution in a previously synthesized plasmid that was homologous to the anticipated cut site in the WIT region, essentially allowing the Cas9 enzyme to cut at a specific site. The guide RNA and homology-directed repair template were then analyzed by sequencing. A chromatogram is pictured on the top right that visually represents confirmed base pairs of each construct. By cross-referencing these to the published Drosophila genome, we ensured that no spontaneous mutations occurred in these plasmids. After confirmation, both the guide RNA and the HDR template were injected into a fly that expressed Cas9. To ensure that CRISPR-mediated homology-directed repair actually occurred, the DS-RED selector, which is not normally expressed in flies, generated red fluorescent-eyed flies. This allowed us to visually select which flies actually underwent CRISPR-mediated homology-directed repair and which did not. So the next steps involve generating yet another plasmid, called the @B plasmid, which will then be placed into the WIT gene at the @P scar by recombination. This is what will allow for generation of the always and non-phosphorylatable WIT versions. Then we will count the number of boutons in these mutants to determine if significant synapse overgrowth or undergrowth is present, with or without tricornered. I'd like to acknowledge the Collar and Weaver Summer Research Fund, John Carroll University Biology Department, the Breuer Lab at Case Western Reserve University, the National Science Foundation, and Caitlin Totoret. Thank you.